In this episode, we speak with Andrei Georgescu, co-founder and CEO of Vivodyne, a biotechnology company accelerating drug discovery by testing treatments on lab-grown 3D human tissues. Andrei co-founded Vivodyne in 2020 after recognizing the limitations of animal testing in predicting human outcomes. He brings deep expertise in tissue engineering, robotics, and AI, with prior work that includes sending lab-grown tissues into space with NASA and developing DNA synthesis chips. Vivodyne's platform integrates human tissue engineering, scalable robotics, and advanced machine learning to generate predictive human data, reshaping the future of drug development. I'm your host, RJ Lumba. We hope you enjoy the show. If you like the episode, click to follow.
RJ Lumba is the executive chairman of Market Insight Media and the managing partner of GrowthCap. He is the host of Great Entrepreneurs, a podcast for driven entrepreneurs and business owners. Our podcast covers all topics related to the entrepreneurial journey, from ideation to execution to optimization and to reaping the rewards of a well-run business. In the minutes ahead, we'll share with you unique insights and tactics from some of today's most accomplished and inspiring entrepreneurs.
Andrei, thank you so much for taking the time to chat with us. It's a delight to be with you.
Sure. Thank you so much for having me.
So very interesting background, very focused on biomedical engineering, and you've come up with a super fascinating technology or solution to clinical trials. And maybe to start off, tell us about yourself, and then we'll head right into the company.
Sure. So my technical and scientific background is in the bioengineering of lab-grown human tissues. And it spanned figuring out the ways to do that really at scale and the ways to do it consistently and the ways to make sure that patient-specific variability and genetic backgrounds and demographic differences shape differences in those tissues that we grow. So from that scientific side, all the way through to the ability to really deploy these at scale with the robotics and software and control systems and all that wrap around those. And so spent my PhD and been time before working on those things, and then spun off Vivodyne as a product of that research.
So what was the moment where you realized this was the idea to really go after?
The really big wow moment for me was when I started grad school. I had never really been exposed to really deep biology and physiology. I was doing bioengineering, but it was more of like the engineering side, right? We were doing DNA synthesis platforms and biosensors for this and that. It was never really peeling back like biology itself. And I remember on one of the first days of my rotation, actually in Dan's lab, looking through a microscope and just seeing what was then a very early kind of evolution of this approach of growing these tissues, but still an extremely striking one, right? Just being able to look through the kind of eyepiece of a microscope and see in front of me this thing that looked a lot like a tissue, but it was growing in a piece of plastic. And I remember thinking back, I kind of laugh at like the naivety of this thought, but I remember having, it was like a small existential, like, why does biology even need us, right? Why do our cells even, like need me if they can grow this tissue in a little piece of plastic? It was my little thing, but that really opened my eyes to thinking, I've never seen anything like this before. And if this capability exists, then we need to figure out how to grow it and how to amplify it and just scale it up to being a powerhouse of how we do just investigations and research on human physiology and biology, right? And the kind of discovery and development of drugs as the primary mechanism that we have to perturb biology in the ways that we want, right? To solve disease. And so, from the very beginning, a week later, Dan and I were planning what the next 4 years of my work would look like toward this goal of this has to be a scaled up approach where we are collecting massive amounts of data at scale. And here's how we commercialize it. And, we were like setting the path of Vivodyne from the very start of that.
And the world at large is familiar with the idea of testing on animals before heading into human trials and proceeding forth beyond that. And so is it really the case that it's not that predictive to test in animals, that there's still a lot of like margin for error is pretty wide testing with animals?
Well, I think a really helpful way to think about it is it's like we are just fine tuning on the wrong thing. And to really generalize this, and I'll kind of explain the animal testing portion, it is like building, an app or a product, but you've tested on the wrong users, right? Like you have the wrong feedback. And so the way that this manifests here, humans and mice, let's say as comparison, we are both mammals, right? We have a pretty much an equal amount of complexity. Like the mouse is definitely smaller, but that does not mean it is less complex, right? All of the ways that its cells talk to one another, is the same. It's like saying, is a baby less complex than a full-grown adult? The answer is no. And so same amount of complexity between humans and mice, both mammals, but we can test on the mice. And so what we find during the development and, earlier than that, the discovery of drugs is when we test on animals, this fundamental approach that we can make a drug molecule that can correctly change the physiology of that mouse to recover from some disease, right? Or to prevent the disease from developing, or to make it live twice as long, or to do all these miracles that we see all the time and routinely in mice. The scientific intervention that we take in making that therapeutic works on the mice. And so the kind of reason for failure of these drugs going into clinical trials at a rate of 95%, right? Like 19 out of 20 fail, is not because the fundamental approach of making a drug is flawed, but because we are optimizing and fine-tuning that drug on mice, and the accumulation of all the small differences that make us human and a mouse, even though we're both mammals, also serve to undermine how that drug then performs in humans, right? And so the solution to this is, well, instead of just fine-tuning and testing on the mice, why don't we just test in humans instead? But for obvious ethical reasons, we can't just go and experiment on like the human experimentation. And so at Vivodyne, the approach that we've taken is, instead of testing on a person, what if we can just grow their organ tissues and little pieces of all of them and grow them to the point of having like thousands and thousands of twins of that patient, and just test every possible combination. And then instead of just one patient, what if we can encompass thousands and thousands of different patients? And this gives us the opportunity to perform human testing before actual clinical trials on people, so that these drugs that we optimize are optimized for human biology and not for animal biology.
And if I were to read the press release, it looks like what you're doing is you're growing these tissues, human tissues. And so one would think like, well, that's not the full human anatomy, right? Like, how would you test to the extent that you have a whole system that you can see how a drug runs through the whole system and what its implications are? But you're hinting at that there's all these organs that you can produce. What are the limitations?
Yeah. So the way that we incorporate this connectedness of all the different tissue types is that we can physically connect them together. So each of these tissues is perfusible through a blood vessel network that forms in each one, and we can just link those together, just like the different organs in a person. And these blood vessel networks, they're like created by the tissues, right? They form just like they would in development. You have blood vessels, you have cells from the immune system, the nervous system, the functional epithelium of the tissue, all of those form. And then the same way that we have systemic circulation in people, we can just connect them together through this living vasculature in these lab-grown versions. But the kind of domains of things that we're not so great at solving yet, I would say, as a simple way to think about it is, imagine that I break my hip or I break my femur, and I need to be in a cast for a while, or I get a hip replacement, and that changes the way that I walk. So I'm putting different pressure on my knee joint, and that makes my cartilage kind of degrade at a different rate, right? And I start to get arthritis in my knee. That's a very mechanical thing, right? And so if I had a little piece of knee tissue, I'm still not putting the weight of a person on it. I'm still not mimicking how the gait would change. And so these kind of more like mechanical matters, I break a bone and my gait changes, or I have heart valve regurgitation, or it's not a dysfunction of cells themselves, like fibrosis or cancer or autoimmunity, or these things that are kind of more systemic. Instead, it is a specific mechanical thing. Like I am loading my knee joint the wrong way, or I've sprained my ankle, or that sort of domain. You need a human-sized model for that. Pigs and some of these things are used currently. And then the other domain that I think we're still a little bit of ways from is the domain of like psychiatrics and mental health and so on. And so things like PTSD and depression, you need a full brain, right? And so that's like one of the things that makes this approach ethical, is we can grow a piece of liver, a piece of kidney, a piece of bone and intestine and heart without it being a person with a brain that thinks, oh, like you're testing on my liver. And so I think it'll be a while before we have a brain in a jar.
And so now the use of proceeds from this latest round is going towards building a facility. It sounds like you're going to have square footage. If I kind of picture this in my mind, I'm thinking like you're going to have each of these replicas, small replicas of like a human system. Is that the right way? And how big is each replica?
So each tissue, the way that you can kind of imagine this is, it is like a very, very large array of individual pieces of each tissue. And we manufacture the microfluidics that house each chamber that will grow a tissue of a certain type. And then when we go to test the effect of, let's say, some developed drug that is a candidate for clinical trials, or we do very early discovery in functional genomics and we try to find new targets for drugs, we grow those tissues kind of just in time. So they take about a week or two to develop until they're mature. We kind of accelerate the process. And it kind of matches, as an interesting little side note, it matches the time that it takes for tissue to heal because we're using the same kind of self-assembly properties that healing tissue also uses, right? To kind of self-assemble itself back into a healthy state. But you grow them just in time over this 1 or 2 week period. And then following that, we immediately test on those tissues. And so they're not like, we don't have these big fridges and freezers where we just keep trays of these tissues. We always grow them as soon as they're needed for an experiment. And so you can imagine machines that are like about the footprint of a desk and about 9 feet tall, full of, robotic arms and fridges and freezers. And they each have these super large microscopes to let us see, laser microscopes, let us see into these tissues and see what's happening. And we just have almost like data center, like arrays of those large machines that can each grow tens of thousands of these tissues.
Mm-hmm. And within each one of those cylinders or whatever shape they're
rectangular kind of, yeah.
Is there like all the tissues combined in one system?
It depends on the study. Yeah. So extremely modular in how we do it. So we can grow many, many different types of tissues, and we can grow many of the same tissue from different donors, or, many different types of tissues from many different donors. One of the really powerful things is when you're looking at combinations of drugs, for example, right? Combinations of drug candidates, the combination space is huge. Like there's so many combinations that might be the key to working well, and then many others that might not be. And so the ability to test, let's say you have 3 classes of drugs with 10 constituents each, and you have 3 concentrations of each of those, you very quickly get into the thousands and thousands of potential combinations, right, to screen through. Like you can't even test on mice at that scale, right? And so, you can subsample and maybe do a little bit of that, but you're just shooting the dart at the dartboard and saying, I think through intuition that this is the right answer, but it's very hard to be accurate. And so the ability to test on all of those things at scale also kind of shapes how we use these. What helps a lot is that we're growing them like right as we test on them, right? And so we can be extremely study specific. We can look at a whole bunch of lung tissue if we're looking at lung fibrosis as an example, or if we're looking at the safety of a drug that we already know is going to be effective on those lung tissues, we can grow our entire portfolio of all the other types of tissues for many different patients and say, what could the side effects of this drug candidate be across these different tissues?
What would be the predictive rate if animal testing is at 5% success rate or predictive rate? What is it with your human tissues?
So what we have seen so far, the fundamental prediction rate will be in this 50 to 100% range. And so at least a tenfold improvement in what we're seeing already in the realms of extremely complex diseases. So like you have these chronic diseases like fibrosis and liver disease, autoimmunity, the extent to which our findings match what is available clinically, even in cases where we don't see it. So if it's proprietary information from a pharma and we are doing this experiment, not knowing that clinical outcome that they have, and then comparing retroactively, after we've done our predictions, I have an immense amount of conviction based on our success in doing those comparisons. And so I think the areas that will be the kind of last pieces to go where we have low kind of prediction is these cases of like, I don't have any real disease of the heart, but it's a anatomical defect, like my heart valve is slightly leaky, or have a hole between the ventricles, or I broke a joint and it's causing this kind of strange, symptom as a result. That's the area where we're not going to be predictive just by the nature of what we're looking at. And it gives us an opportunity to grow in that domain as well.
Mm-hmm. Where are you now in terms of the stage of the company? Have you started working with some companies?
Yeah, we're working with a majority of the top 10 pharma already. And the really interesting thing, I think, is we've engaged like throughout the preclinical pipeline, both in, pipeline spanning partnerships, but also in super, super high, like we have a drug and we just noticed that another drug that was also trying to get to market failed in this strange way in safety. And we are now worried that if we bring ours to clinical trials, it might fail in that way. Can we do a safety assessment in, of our chemotherapeutic in the bone marrow? Or is this molecule going to be immunogenic in some types of patients? And so we spent everything from discovery and validation very early in the pipeline all the way through to the distribution of the drug, the uptake of a drug. Is there going to be immune side effects? Are we going to have immunosuppression, myelotoxicity risk, and through all of those? And so, it's like the breadth and like the number of tissues this has spanned all, I think, are really, really important conviction builders for me personally, right? Like this has to be worth it, right? And so we want to build a great business in all of this. But fundamentally, I think everyone here at Vivodyne is doing it because of the potential for really changing how medicine is developed. So far, we're very pumped.
We're coming up on the tail end of the conversation. I have two final questions. One is, can you tell us about a person who has had a profound influence on you?
There are a couple. First is Richard Feynman, and his approach to understanding physics from first principles has been a huge influence in the way that I approach like how stuff around us just works, right? From a first principles perspective, I think I have an immense amount of curiosity about how did we decide on the form factor of that power outlet? Why is it designed that way? How are those connections insulated? It's like everything around us is designed by people, and there's often very informative reasons for the way something is the way it is. And ultimately, it kind of falls down to the fundamentals of physics, and that Feynman approach of taking it from first principles has really shaped the depth that I like to go to in understanding this. Interestingly, I think like the exception to all this is, right, we've designed everything around us except ourselves. And I think we as people should still kind of maintain some humility, but we have not seen anything in the universe that is as complex as, let's just say, life as a kind of hand wave, but even within life, the human brain, right, is about as complex as we've seen, and we did not make it. And so the ability to learn the way that evolution has shaped it is like yet another, you can do this whole cycle from the very top to bottom, all the way down to first principles on something that was not human engineered. And I find that to be this super fascinating domain in biology.
Last question. Can you tell us about a charity cause or other endeavor that you're passionate about?
I think that the biggest thing that really motivates me in this area, one is, I think the science is cool, but I think, building robots and all this stuff, I love every day. But the really big driver is, it seems like just in general, it seems like all of the technologies that people are building seem to be moving at a really rapid rate. So there's satellites just flying above us to give us GPS. There's, self-driving cars that I can talk to my computer now, right, in a way that I was not able to, even unforeseeably, right, 2 years ago. We have made all these strides in technology, and yet in medicine, like the first line treatment for some cancers has not changed since the 80s. And I think that is a huge, I don't want to say failure of it, because I include myself in that group of people responsible for trying to find a solution, right? But it seems so strange that we have not really made any great strides in that domain. And so all of my focus and all of my kind of energy and passion is directed at this domain of what is fundamentally stopping us from making drugs in a very revolutionary, like what is going to change the regime of how drugs are developed fundamentally, right? To change it from what seems to be unchanged from the 80s, apart from a couple new modalities. But the regime itself, what will it take to change that is the target of my focus. And I think that fundamentally is to change the substrate on which we do our testing. And it should not be rooms and rooms of mice in cages or, primates and dogs in cages. It should be bioengineered human tissue that is translatable directly to those human patients that will be taking these drugs that we develop. And so, yeah, I am super locked in on that.
Excellent. Well, Andrei, I want to thank you again for taking the time to chat with us today. It's been a wonderful conversation.
Yeah, thank you so much for having me. I really appreciate it.